So now we can discuss about the topic that is primary and secondary standards, which is an important topic and basic term that is used in the pharmaceutical analytical chemistry. So what is called standard solutions? Standard solutions are the solutions of accurately known strength, and they are having a very much importance in the analytical field. And this can be prepared by taking a known weight of the reagent of highly purity in a definite volume of the solution. Next, functions of the standard solution. They are mainly used to provide a reference using which we can determine unknown concentration of the solution, and they are mainly used to standardization of volumetric solutions, used for the preparation of secondary standard, and to calibrate an instrument. Then there are two types of standard solution. The first one is the primary standard and second one is the secondary standard. We can discuss in detail what is primary standard and what is the secondary standard along with their properties and examples. So first is primary standards. Its definition is they are the substances that is having a high purity, and the purity range is between 99.95 to 100.05% with respect to the active component, that should be stable at our temperatures for drying, used to standardize the volumetric reagents. So primary standards are the standards that are mainly used to standardize the volumetric reagents. They are having a high purity and the purity range is 99.95 to 100.05%, and they have to withstand the drying temperature. That means they should be stable. And these are the substances which are available in pure form with definite chemical compositions, and these substances are called primary standards. Then next, what are the requirements or what are the properties of the primary standard? The first property is it must be easy to obtain, to purify, to dry and to preserve in a pure state. It must be easy to obtain means easy procurement, that means it should be cheaply available, and to purify means the pure form should be available or it should be easy to purify. And to dry means the drying temperature of the almost all the primary standards are between 110 to 120 degrees Celsius, and they should be able to preserve in a pure state. And based upon this property, this requirement is not suitable for the hydrated substances, because hydrated substances, they may contain water molecule or water content will be present in the hydrated substances, and because of this it will be very difficult to remove the surface moisture. Second property is the substances should be unaltered in air during weighing. Unaltered in air during weighing means the substances should be stable. They are stable in the sense it should not be hygroscopic. Hygroscopic means it should not contain, it should not absorb water. And next is it should be oxidized. It should not be oxidized by the air, or it should not be affected by the carbon dioxide, and this should be unaltered during the storage conditions. This indicates that the substances should be stable. And the third one is the substances should be capable of being tested for impurities by qualitative and other tests of non-sensitivity. And during this testing, the total amount of impurity should not exceed 0.01 to 0.02%. And the fourth one is it should be having a high relative molecular mass so that we can avoid various weighing errors. And the precisions in the weighing is ordinarily 0.1 to 0.2 milligram for an accuracy of 1 part in 1000. And the substances should be readily soluble under the conditions in which it is employed. That means they should be readily soluble in the solvent in suitable volume and the suitable atmospheric conditions. And the sixth one is the reaction with the standard solution should be stoichiometric and practically instantaneous, so that almost all the titration errors can be avoided, and it should be easy to determine accurately by the experiment. Now we can move on to the examples of the primary standard. So the primary standards that is used in the acid-base titrations are The acid-base titrations can be classified into alkalimetry and acidimetry. Alkalimetry is the titration with the sodium hydroxide, and acidimetry is with the hydrochloric acid or sulfuric acid. In alkalimetry, the primary standard used is the potassium hydrogen phthalate. In acidimetry, the primary standard used is the anhydrous sodium carbonate. And in case of oxidation reduction titration, it is also called a redox titration. That is, in permanganometry is the titrations with the potassium permanganate. The primary standards used are arsenic trioxide, sodium oxalate and potassium dichromate. And in case of iodimetry, the iodometry is the titrations that is along with the titrate will be the sodium thiosulfate, and the primary standard is potassium bromate, potassium dichromate and potassium iodate. In case of iodimetry, titration with the iodine, the primary standard is arsenic trioxide, and this same will be the standard for the seriometry, that is the titration with the ceric ammonium sulfate. We are using the primary standard as arsenic trioxide. Then precipitation titrations, example Morse method, the primary standard is the sodium chloride, and in case of complexometric titrations of granulated zinc and disodium edictate, the primary standard is calcium carbonate and magnesium sulfate. In non-aqueous titrations, perchloric acid, we can use potassium hydrogen phthalate as a primary standard. In case of sodium methoxide, lithium methoxide and tetrabutyl ammonium hydroxide, the primary standard is benzoic acid. Next, we can discuss one important properties of the primary standard. The primary standard, we cannot use any hydrated salt because they are having a tendency to lose a varying amount of water on drying. We have already discussed that the primary standard have to be dried at 110 to 120 degrees Celsius. So in case of hydrated salt, if they lose water content, means they cannot be used as a primary standard. Because of this difficulty of efficient drying, hydrated salts are not preferred as a primary standard, and this can be explained with the help of the example sodium hydroxide. The sodium hydroxide, since they are having a hygroscopic property, they can be standardized by titrating against a standard solution of potassium hydrogen phthalate or against a standard solution of hydrochloric acid. Here potassium hydrogen phthalate is a primary standard, solution of hydrochloric acid is a secondary standard. Now we can discuss what is secondary standard. The definition of secondary standard is, it is a solution which contains exactly known amount of the substance in unit volume of the solution, and this can be determined by titrating against a primary standard. That is the difference. The secondary standard can be determined by titrating against a primary standard. And another important point is, secondary standard is a substances which for one or more of the reason cannot be used as a primary standard. For example, sodium hydroxide cannot be used as a primary standard because they have the tendency to absorb water and carbon dioxide from the atmosphere, and the composition of the solution is subjected to wide variations at different periods. Then the properties of the secondary standard are, it has less purity, they are less stable and hence more reactive, but its solution remains stable for a long time and can be titrated against primary standard. Examples are, anhydrous sodium hydroxide is a secondary standard. It is highly hygroscopic. Potassium permanganate is another compound that is often used as secondary standard. It is less stable and is reactive. Therefore, when potassium permanganate solution is prepared, it has to be standardized with a primary standard. Then I have prepared a table, that is comparison table between the primary and secondary standard solutions. In case of definition, the primary standard is a solution made from primary standard substances, and in case of secondary standard, they are made from secondary standard substances. Purity, the primary standard is highly pure and the secondary standard is low pure when compared to that of the primary standard. In case of reactivity, primary standard is very low reactive and the secondary standard is high reactive. In case of nature, the primary standard should not be hygroscopic, secondary standard can be hygroscopic. In case of contamination, they are rare. Primary standards are rarely gets contaminated, while secondary standards are easily contaminated because they are highly reactive. and in case of applications they are used to standardize the secondary standards and other reagents. While the secondary standards are used or they are very much important for the specific. And thank you all.